Yes. So, if you remember, we were talking about this biotechnology, right? And in the previous sessions, we saw that how you could take this gene and you could put it into a particular cell, right? And I told that there were different vectors which were used for it. We saw that there were enzymes which were cutting it. That was your restriction endonucleases, right? And then it produced those two types of ends, you remember, the blunt ends and the sticky ends, right? And then there was that insertional inactivation which helped you to identify that whether your gene of interest has actually gone into what you call as the vector, and whether the vector has carried that gene to your host, right? Today we'll be talking about the next step in that, that is preparation of a competent host for transformation with your rDNA. See, now I've prepared a rDNA. I've prepared a recombinant DNA, a DNA which has what you call as segments from different organisms, that is called as a recombinant DNA, right? So now I have a recombinant DNA, and now I'm going to put this recombinant DNA into the host, right? So that is what is called as transformation. When you transform something, that is transformation. So when this host is going to get a DNA from outside, probably this host will show new characters, and we say that this host is now transformed, it has changed, right? So here we are going to prepare a competent host for transformation with your rDNA. Now DNA, basically, if you see, it is a charged molecule, it has a negative charge, so it is hydrophilic. That means it loves water. It cannot cross the membrane easily, right? Because the membrane, it is mostly hydrophobic. And then you'll find that there are also cell walls and other stuff if you are trying to put it in a plant cell or even in a bacterial cell, right? So there is a major basic barrier. So DNA being hydrophilic, it cannot pass into the host cell. And host cell is not like ready to accept a DNA from outside. No one is going to accept a DNA which is coming from outside. So you need to prepare that cell in such a condition that it will now accept that DNA. So preparing that cell is called as the cell which is ready to accept the DNA is called as a competent cell. So I want to prepare a bacterial cell that is competent to take up this extra DNA. And this, I tell you, can be done by treating that cell with a specific ion, which is a divalent cation, and especially that is calcium. So if you're growing this bacterial cell in an environment which has a lot of calcium, right, probably it increases the efficiency of that bacterial cell to take this foreign DNA. Otherwise, it will not, right? And like not only calcium, but now you need to introduce some holes in this bacterial cell so that the DNA can actually easily go. And that is done by using a heat shock method, right? Where you take this particular cell, what you call as host cell, you take this recombinant DNA, you have put it in a solution which contains a lot of calcium, and now you have kept this on ice. Now you keep it on ice for some time. Now once you have kept it on ice, after some time now you will transfer it for a few seconds at 42 degrees Celsius directly. So now it was on ice and now it comes at 42. Now you take it at 42, again you immediately take it and put it on ice. So you see there is a variation in the temperature. So temperature is very low and again there is very high. That is called as a heat shock. So it is in the ice, you give it a heat shock, again bring it on ice. So that creates holes in the membrane, right? So the recombinant DNA is forced by incubating the cells with this recombinant DNA on the ice. And now you take the temperature to how much? 42. That is what is called as a heat shock. And then you back take it on the ice. So bacteria will take up that DNA. Right, so that is one way of making or transforming your host cell, but it's not always like done in the same way. Now there are many other methods also, like they go for what is called as a micro injection. So micro injections, like you can look under the microscope and like you can have a very small injection which is actually putting that piece of DNA into the cell. You can actually manipulate that. That is called as a micro manipulator. Like आपका डॉक्टर कैसे इंजेक्शन लगाता है तो he sees where the vein is or where he can actually put that. So when you actually put that needle into a proper vein, the thing goes in, right? So here what you're going to do is you're going to inject this recombinant DNA. I'm going to fill that recombinant DNA. I'm going to target that particular cell and I'm going to actually put that as an injection into the cell. So now it's directly inside the cell. That is what is called as what? Micro injection. Another way by which it is done is this biolistic or gene gun. You have seen, you go to some yatras and jatras and then you have those what you call as balloons out there, and you have that gun, and with you can fire that, right? So what you do is there are small those गोलियां रहती ना ऐसे गन में, और फिर उससे आप उसको टारगेट कर रहे हैं, फिर आप हिट कर रहे हैं तो बलून फूट रहे हैं, राइट? तो लाइक एज यू टारगेट, तो व्हाट यू आर गोइंग टू डू इज नाउ यू वांट टू टारगेट योर सेल. So you have a bacterial cell, you have a plant cell, especially this is done with those plant cells and all. So you have that plant cell. Now you have your DNA. Your aim is to put this DNA back into that plant cell. So I have that DNA, and that DNA is coated on what you call as the tungsten particle or the gold particle. So I take a gold particle and I apply the DNA which I want to insert into that on that gold particle, and now with the biolistics or this gene gun method, I'm going to what you call as target that host cell. So when it will hit that host cell, it will go inside that host cell and it will deliver that DNA inside. So when I'm injecting it, that is what is called as micro injection, and when I'm like shooting it, that is called as biolistic or gene gun. So in biolistic or gene gun, I'm going to use the high velocity those particles and bombard it with that, and those particles are going to be out of gold or tungsten, right? Then you can also use a disarmed pathogen. You have a vector, you have an organism which is actually going to infect that plant cell or animal cell. So what you do is you take that pathogen and you remove the piece of DNA from there, you put your piece of interest there, and then when it is infecting that plant or animal, it will take your gene of interest there and it will go there. So you can either give a heat shock. Heat shock was on ice, then at 42, then again on ice, which created holes. Micro injection, coating them was biolistic or gene gun, and that coating was done with either gold or tungsten, and then the disarmed pathogen. So basically, how do I do this recombinant DNA technology? When I'm doing this recombinant DNA technology, what I'll do, what are the basic steps? Last to last need, there was this sequence of steps they have asked that how you're going to do it. So first thing which you're going to do is isolate the DNA. The DNA which I want to what you call as amplify. Like if I want to produce insulin, I'll need a human cell, I'll need a pancreatic cell, and from that I'm going to isolate the DNA. So first is isolating the DNA, then fragmenting the DNA. So first step will be like I'll have a bacterial cell, right, or a host cell or a target cell or whatever, like from where I'm actually isolating the DNA. Like from insulin, it will be a beta cells of islets of Langerhans of pancreas. I'll take that cell, I'll isolate its DNA. Now from this DNA, I'll make its fragments. Right, what I did is I fragmented the DNA with the help of restriction endonuclease, obviously. And then I'm going to isolate my gene of interest. I want only that gene from that entire cell which is actually coding for insulin. So next step would be isolating the desired gene, that DNA fragment. Then I'll take a vector, say like a plasmid. So I'll ligate that to the vector, transfer it into the host cell. Now that I'll put into the host cell. See, E. coli is a host cell. So now I'll transfer it into the host cell and now culture this host cell on a large scale. So I'll now produce many such bacteria on a large scale, and these bacteria are now producing insulin for me. So what was the first step? Isolating the DNA. The next step was fragmentation with restriction endonuclease. Then out of all those fragments, I isolated my gene of interest. Then I took this gene of interest, put it into the vector, and then I put this vector into the host, right? And then I put this the recombinant host into a proper culture medium, so that I get multiple copies of this cell, and so I'll get multiple copies of the gene or of that product, right? So that is what how recombinant DNA technology is done. So I'll grow those E. coli cells in a huge vessel where I can control all the parameters, and I'll get a lot of insulin from there, right? So that is how it is done. So the first step, as you saw, was isolation of your genetic material. You want to isolate that DNA from the given sample, right? So basically we all know that when it is a eukaryotic cell, DNA is present inside the nucleus. You're not going to just go and say the cell mala DNA there. It's not going to give you the DNA like that, right? You'll need to break open that cell. And now when you're going to break open that cell, remember all the debris is going to come out. It's not only the DNA that will come out. Like you're not going to airport, like you're not holding a station there ki where you come, so only DNA will come to you. As soon as you open the cell, what will happen is everything will come out. And from that you want to purify your DNA. So the first and foremost thing is like you need to isolate the genetic material. Fortunately, in most of the organisms, like it is DNA that is genetic material. Like some RNA, like some viruses have RNA as genetic material, but for most of the other cases, it is the DNA. So you will take the DNA, like you're going to cut it with restriction in endonuclease, but then for the first thing, first priority which you'll have is that it should be pure. You're not going to take an impure DNA and put restriction endonuclease. It will not do the work, right? So you need the DNA in the purest form. So you need to break the cell to release the DNA. As I told, when you break open the cell, the carbohydrates, the proteins, the lipids, the nucleic acids, the RNA, everything will come out. And what you are interested is only the DNA, right? You don't need anything else, right? Again, to break open also, depending on which cell you are using. Like if I'm using a bacterial cell, probably I'll go with lysozyme, which will break the cell wall of bacteria, which is made of peptidoglycan. If I'm using a plant cell, probably I'll go with cellulase, which will digest the cellulose, because the plant cell wall is made of cellulose. Like if I'm going with fungus, I'll use chitinase, because we know that fungal cell wall is made of chitin. So if it is bacteria, it is lysozyme. If it is fungi, it is chitinase. If it is plant, it is the cellulase, right? So what I'm going to do is I'm going to use these specific enzymes. It makes the reaction more specific. To remove other things, like I'm going to use specific enzymes again. Like if I want to remove protein, I'll use protease. It will degrade all the protein. If I'm going to remove, say, RNA, I'll add RNAs in that. So RNAs will destroy all the RNA. So basically DNA will come out with some proteins, that is histone proteins. If it is a eukaryotic cell, because we know that eukaryotic cell has DNA complex with those histone proteins. So when you're going to break it out, what will happen is you need to remove all this debris. So to remove proteins, you will have to add protease. It will be complex with RNA because it is undergoing transcription. So you will have many places where DNA is complex with RNA. So you may add RNAs, which will degrade the RNA. And now you want a pure DNA, and for that you're going to add chilled ethanol. So at the last step, now we are not going to see step by step how the DNA is isolated, but then at the last step, when you have added all these chemicals to other stuff to remove all of the debris, you have to be very careful that in the entire process DNA doesn't get damaged. I can break open a cell by adding a lot of acids, but that will damage my cell. I can break open a cell by just heating it, but that will denature my DNA, right? I don't do that. So I have to be very smooth and I have to be very sober on the cell so that everything gets destroyed, but the DNA remains as it is. So when I'm precipitating the DNA, for precipitation of DNA, what I'm adding is the chilled ethanol. So addition of chilled ethanol will precipitate my DNA. And once that DNA is precipitated, I tell you, I can literally see it as fine threads. Some can actually spool it. If you add a glass rod or something like that, you can literally spool the DNA. So after addition of a chilled ethanol, you'll find that DNA, like, it can be spooled in the form of fine threads. It precipitates out. So what you have to remember here is addition of chilled ethanol, it precipitates the DNA, right? So basically, once you get that purified DNA, now you're going to cut the DNA at the specific location. And I said that you're going to do it with what is called as restriction endonuclease. So I'll take the DNA and I'll incubate it with restriction endonuclease. When I'm incubating with restriction endonuclease under its optimal conditions, like whatever temperature it requires, whatever pH it requires, whatever other factors it requires, I'm going to provide it that. And once it is cleaved, I'm going to perform this agarose gel electrophoresis. You remember we had seen that in agarose gel electrophoresis, you prepare agarose gel and then you place your what you call as fragments here in the well, and the DNA which is there, which is negatively charged, it migrates towards the positive charge. You remember in the last lectures we had seen. According to their sizes, they can actually move. So you can actually monitor that how much fragmentation has been done. So the DNA will start moving. DNA being negatively charged, it will move towards the anode. You can do a similar thing with the vector. And you remember vector is the one which is going to actually carry over this DNA. So you take the DNA, isolate it, cut it with restriction endonuclease. You take the vector, cut it with restriction endonuclease, and then now you can join this vector to your gene of interest. So you can mix it and you can add what is called as ligase, and then what you get is this recombinant DNA. It looks like a strawberry, but it is not like that. So it is your DNA molecule with your gene of interest. Again, another DNA molecule. So say this is a plasmid, right? And this is your what is called as gene of interest, right? If you remember in our earlier lectures also, we had seen that in DNA fingerprinting, or even here, you see that you require to work with the DNA a lot. You require a lot of DNA. right? And when you're isolating the DNA, like what do you think, that you will get a lot of DNA? You're going to get DNA kilograms or what? No, you're going to get DNA in a very less quantity, in micrograms or milligrams and nanograms, right? So the basic step in biotechnology is amplification of this DNA. So you have got the DNA now, but you're going to amplify this DNA, right? So this amplification of DNA, which is done, that is done with a special technique which we call as PCR, called as polymerase chain reaction, right? So this particular technique of PCR, that is polymerase chain reaction, can get you billions of copies of your DNA within few minutes, within few hours, right? Because you're going to do a in vitro amplification. So PCR is a in vitro method of gene amplification. So basically PCR, polymerase chain what? Reaction. Polymerase chain reaction is used for what I told, for amplification of your DNA. So you need multiple copies of your DNA, right? So what you're going to do basically is, you're going to take your DNA and you're going to put it under various cycles of temperature. Like you will take the temperature to 90, 94, something like that. Then you'll bring the temperature to around say 50, something like that. Then you will bring the temperature to around 70. So this thermal cycling goes on. Now if I raise the temperature to 94, what will happen is, say about 90, if I raise the temperature, the two strands of DNA, they get denatured, right? So they get separated. The two strands of DNA, they get separated. Now what I do is, here I add primers. I add what are called as the primers, the forward and the reverse primers. So I'll add a primer here, I'll add a primer here, the forward and the reverse primer. And then when the temperature again goes around 70 or something like that, now whatever primers I had added, they get extended. Oh, and now ultimately, if you look at the result, I got 2 copies of my DNA. Remember, we had seen that there was a semi-conservative mode of replication, one old and one new. So I started with one molecule of DNA and I have how many? 2. And this, if I keep on repeating, I can have around billions of copies of DNA in only 30 cycles of such repeat. So that is done in PCR. So what is required in PCR? The gene which I want to amplify. So this is the gene which I am going to amplify, which I need multiple copies of. What I want to need is the primer, which is going to start the replication. You remember, DNA polymerase cannot start the replication. So I need primers. Then what I'll need, the one which extends it, I need DNA polymerase. Now the problem is that this DNA polymerase has to overcome all these temperatures. The temperature is going to around 90, then 70, then 50. It's like fluctuating. So normal DNA polymerase probably will get denatured. I cannot use that. So I use a DNA polymerase which we get from a bacteria called as Thermus aquaticus. So there is a bacteria called as Thermus aquaticus. I'm going to use that bacterial DNA polymerase, the Taq polymerase it is called as. Which polymerase? Taq polymerase. So Taq polymerase. Why I have used a Taq polymerase? Because it is thermostable. Even when the temperature goes 90, I do not open and remove. I cannot do that. I just prepare a reaction mixture. Like I'll prepare a reaction mixture. In this, I'll add my gene of interest. I'll add the primer. I'll add the Taq polymerase. I'll add all the nucleotides. Because when it is synthesizing, it is going to need the nucleotides. When it is going to add A in front of T or G in front of C, it's going to add all those deoxyribonucleotides. So I'll add everything in this. आप ओवन में कैसे रख देते, एक बार लगा कि you set a program, इतने मिनट इतना टेम्परेचर, तो वैसे I'm going to put this in that thermal cycle, small machine, I'm going to put it there and I'm going to set the program 90 डिग्री पे इतना देर, 70 पे इतना देर, 50 पे इतना देर, और ये ऐसे 50 साइकिल चलाओ, 20 साइकिल चलाओ, 30 साइकिल चलाओ. So all these steps will happen one after the other and will give you multiple copies of the DNA, right? So in PCR, I'll need the gene of interest, I'll need the primers, I'll need the DNA polymerase, and not any DNA polymerase. Which DNA polymerase I told? Taq polymerase. Why I'll need Taq polymerase? Because it is thermolabile or thermostable. It is thermostable. And then I'll need those nucleotides. So these are the ingredients. And you see those khana khazana, what are you going to require? So you're going to require the gene, you're going to require the primer. The primer is that short stretch of oligonucleotides which are spanning the ends of the gene which you really want to amplify. And then you are going to require the Taq polymerase. And then the machine of the thermocycle. Once you set it, so you're going to go through these three steps. One is what is called as denaturation, other is what is called as the annealing, and third is what is called as the extension. So you're going to go through those three steps every time. So denaturation, the two strands of DNA get separate. Then annealing, your primer gets attached. And then extension, from the primer you extend it. And now you have how many copies? 2. Now what will happen is in the next cycle, from these 2, it will again undergo denaturation. So from next cycle you will have how many? 4. In the next cycle you will have how many? 8. In the next you will have how many? 16. So the number keeps on doubling. And that is what is called as your PCR, the polymerase chain reaction. So polymerase chain reaction actually increases the number of your copies of DNA. It was the technique which was first introduced by Kary Mullis, but I think your textbook, it doesn't give that. Like if you look what is given in your textbook, it says that PCR is a reaction where you're going to produce multiple copies of your DNA. You need primers, the enzyme, the DNA polymerase, which is going to extend that and process repeated many times, and segment can be amplified billions of times. And this is achieved by a thermostable DNA polymerase from a bacterium which is called as Thermus aquaticus. So it remains active at high temperature even when you induce denaturation of the double-stranded DNA. So the three steps which I showed, right? One was what is called as what? Denaturation, and other was what is called as the annealing, and third was what is called as the extension. So I'm going to what you call as get that particular multiple copies of my DNA, right? If you look at the next sequence which they have talked about, that is insertion of a recombinant DNA into the host cell. So we are going to insert the rDNA into the host, right? So basically just now we saw that I have prepared a DNA which is having this gene of interest, and now this I am going to put into the host cell, right? And now I am going to check whether this has gone into the host cell or not. If you remember, we had seen that multiple antibiotic resistant, ampicillin, tetracycline. Apart from that, now looking in a simpler way, if this vector is having a what you call as gene for ampicillin resistant, and if it gets transferred here, right, if there is a transformation which has taken place, if I add antibiotic, right, I know that this is carrying the gene, therefore it will survive. If there is a bacterium which does not carry that gene, that it will be died. So in this case, that antibiotic resistance gene is said to be working as a selectable marker. I use that as what is called as a selectable marker. So I can select the transformed cells by just checking that whether it has that resistance towards antibiotic. If it has resistance towards antibiotics, I can be sure that my gene of interest has gone in there, or transformation has taken place, the vector has gone in there, right? So that is what is called as the use of selectable marker. You see, they have told there are several methods of preparing this recombinant. You have a gene which is resistant to antibiotic like ampicillin, and you transfer it into the E. coli. So the host cell, it becomes ampicillin what? Resistant. And if I transfer this to a plate containing ampicillin, right, all transformants will grow, whereas the non-transformants, they will die. So I'll prepare a petri dish. It's not something which you're going to eat. It is a nutrient medium which contains all those nutrients, and I'm going to spread those bacteria all there. So that dish itself contains those antibiotics. So if it has the marker gene, it will survive. If it doesn't have the marker gene, it will die. So I can do that, right? I know this is a quick revision, but for more detailed this thing, you can come back to my videos and we can have a more detailed discussion. But now we are preparing in a very quick manner, right? So another thing which they have talked about is obtaining the foreign gene product. Now that is a big challenge. So obtaining your foreign gene product, if I am going to get that product from the bacteria in which I have inserted that gene, you know that I need to grow that bacteria on a large scale. Now when I am growing that bacteria on a large scale, you know that I need to provide it proper conditions. Bacterium will not grow if it is not getting enough oxygen. It will not grow if it is not growing in a medium where the pH is maintained, right? So therefore, you remember in microbes in human welfare, we had seen that there was a big vessel in which you grow all these bacteria and which was called as the fermenter. So similar kind of fermenter I'm going to use here. So what is my basic requirement is that when I'm growing my bacterial cell, a host cell, which is having my foreign gene product, I should maintain all the conditions in the optimum manner, the temperature, the pH. I should see that no other bacteria comes in. So all the conditions which are there, they are kept ambient, right? Optimal, what I say. So proper temperature, proper pressure, proper pH, right? Proper source of nutrients I have to see. Because if the bacteria is like eating the nutrients there and the nutrients get depleted, the bacteria stops growing, my production of the product will also stop. So I need to constantly check whether it requires anything, if it requires any nutrients, right? So I need to constantly supply with those nutrients also sometimes. I need to have a check over what is happening inside, right? So for that, you prepare what is called as your various bioreactors, right? So common bioreactors which they have talked about is a stirred tank bioreactor, right? A typical stirred tank bioreactor like will have a tank, which has those stirrers. You have a typical stirred tank bioreactor. You have some bioreactors which are what you call as having sparge tank bio. You have a provision for providing oxygen there. So that is a simple thing here, but then here you have a provision for oxygen too. So from here you can also provide oxygen to them. So you actually sparge it. So that is called as a sparged stirred tank bioreactor. So you have a bioreactor which is having machines for movement also. So it also has that impeller here which is actually going to move. So here you are going to what you call as have a lot of oxygen bubbles coming here, and then this is a sparger which is going to this is a sparger actually which is giving out those oxygen bubbles, and this is the stirrer which will actually move those bubbles throughout. So this is just stirring. So this is a typical stirred tank, what you call as a bioreactor, where you have a provision for your sterile air, right? Then you have a provision for breaking foam also, right? So there is a foam breaker, because when this will grow, there will be a lot of foam which will be produced. So there is a foam breaker there. You have provisions for acid base, right? You have what they have others showed. You have the motor there, right? You can actually control it, right? So this is the culture broth. So it is moving. So culture broth is like the broth which is containing all those microbes, right? So here you have those bubbles. Actually what happens is when the oxygen is being supplied and then when it is moving, right, what happens is the bubbles they break, and when the bubbles they break, the size of the bubble it decreases, and therefore they have more surface area to interact, right? So these are some of the bioreactors actually which are used to grow these microbes. So you can see there is an acid base balance. So whenever like there is a window also from which you can see, there is a sampling port also from where you can take out the samples from between and check whether the product is being formed. So everything is being monitored, every step. Temperature वाढत असेल, you have a cooling jacket. Temperature कमी होत असेल, so you can heat it, right? You can like if the pH is decreasing, you can add the base so that the pH comes back. If the pH is increasing, you can add the acid so that the pH it decreases, right? You can maintain the pressure, you can maintain the flow, you can maintain how much it is being mixed. So all those things, the controls, they can be made in this reactor. So let us see what your book says, right? So as I told, you have added this alien piece of DNA in this cloning vector. The DNA gets multiplied. So there is a need for the recombinant DNA to be expressed. So what they have told, after the cloned gene of interest has been optimized condition for expression of the target protein, you have to consider producing it on a large scale. अब आपको ये सब कैसे बनाना है on the large scale, and then you are going to put it in a heterologous host, and it is going to produce that recombinant protein. The cells they can be grown on a small scale or on a large scale, right? And then the cells, they can be multiplied in the continuous culture system, right? So it could be a batch or it could be continuous. Continuous is what, like this is going on in a batch. Like I'll have a batch and I'll put the medium, I'll grow them. Once the product is produced, I'll just remove it and I'll use it. In a continuous culture system, what I do is I have a vessel, right? I have an outlet to that vessel and also I have an inlet to that vessel. So I can continuously add nutrients. You can see the bacteria is continuously growing, it is producing the product. I can remove the product from here. So that is a continuous production. So continuous production, you keep on adding the medium and also keep on removing the medium. And that removing the medium, you will have all the product which is being formed. So here, if you look at in your textbooks again, so you can multiply wherein you used medium is drained from one side and you're adding the fresh medium from the other side. So you're maintaining the physiologically most active or the exponential phase and it is producing a larger biomass, obviously. You're keeping the cells continuously active. So therefore the biomass which you will produce and the product which you will produce will be more. If I am growing it in one phase or at a one time, probably the biomass produced would be less, the product produced will be less. It has its own advantages, disadvantages actually. So coming down here, it says that small volumes cannot yield appreciable products. So large quantities require bioreactors. You have a lot of vessels, you have specific products, enzymes. You can use microbial, plant, animal or human cells for growth. A bioreactor, what it has, it will have everything to give it a desired optimum growth, temperature, pH, salts, vitamins, everything. So here you can see a stirred tank bioreactor, a curved base, mixing oxygen availability throughout the reactor, and alternatively in a sparge reactor you can bubble the air. And here you can see that in this sparge reactor you are actually bubbling the air, whereas here you are just mixing it, right? So that care it can be taken. It has an agitation system, or oxygen delivery system, and a foam control system, a temperature control system, pH control system, sample ports, right? Everything can be done. So you can just remove the sample, you can control the pH, you can control the temperature, everything can be done here. You're providing the oxygen, so everything is like automated. And then what comes last is like once my product is produced here, I need to remove my product. See, this broth has a lot of nutrients. You know, it might be having agriculture waste or other stuff in which I am growing the bacteria. Then bacteria is not only producing my product, it is producing so many other products. So I need to remove or purify my product from that. And that particular process of purifying the product from there is what is called as downstream processing, right? So when I am purifying my product, that is what is referred to as what? Downstream processing. So downstream processing is what? Like I'll take this or I'll take this. It is not that small. It is about like thousands of liters sometimes. So I'm going to like remove all that broth and I'm going to filter it out. I remove those bacterial cells if my product is outside the cell. And then I'll have to remove those bacterial cells and I have that broth. This broth has n number of impurities. Now step by step, I want to remove all the impurities. I'll use filtration, I'll use sedimentation, I'll use centrifugation, I'll use chromatography, and then I'll get a finished product, a purified product. And remember that most of these biotechnological products are something which are concerned with your health. So if I give you one product and I say that I'm not sure how much pure it is, but you're going to inject it in your body. If I sell you one product which is of 5,000, I say that I don't guarantee what will happen when it is injected. And if I sell you another product which is 50,000, I say that I guarantee that it has no impurities. Obviously, you will go with 50,000. So biotechnological products, people or as such all people, scientists, they prefer that it should be pure because it should not have any side effects, right? So that process of purifying them, that is what is called as downstream processing. Now actually all the steps of those downstream processing are out of the scope of your book and your syllabus, but if you could like scroll my channel, you can find them also probably, but that is not for NEET, right? But then that is what is called as downstream processing. Let's see what your book says. So downstream processing, after completion of the stage, the product is subjected to series of processes before it is ready for marketing. It includes separation and purification, which is collectively called as downstream processing. So you purify it, right? And then you add some preservatives in it, and then formulations, they undergo clinical trials and then quality control testing. So that what you call as varies from your product to product. कि अभी आपने एक vaccine बनाया, अभी आपने एक antibiotic बनाया, so I'm not going to like go and sell it to people there out. I'll have to take permissions from government agencies. I'll have to do clinical trials on animals, on humans, and only then that will be available. So from product to product, actually, that particular phenomenon of what you call as downstream processing and the trials and permissions, it varies, right? So that was just a summary. Like we just went line by line in the NCERT, like what they were talking about in various steps of biotechnology. So points to remember are that a cell cannot take the DNA directly. The cell which can take the DNA is the competent cell. For that, what you can do is give them shocks, the heat shocks. So for that, you place it on ice at 42, again on ice. You can put it through injections, that is micro injection. You can like shot it, that is biolistics or gene gun. That gun is made of that gold or tungsten. And then you can also go for disarmed pathogens. Then I told various steps in the process where you isolate the DNA, cut the DNA, isolate your gene of interest, take the vector, cut the vector, join it to the vector, put this recombinant DNA into the host, now grow it the host and get your product. For isolating DNA, I told that first you will have to open up the cell. To open up, I told bacterial cell you will use lysozyme, plant cell you use cellulase, and for fungal cell you use chitinase. Then I told that for precipitating the DNA at the last step you use chilled ethanol, right? For removing RNA you add RNase, for removing protein you add protease, and only pure DNA can be used for cutting with restriction endonuclease. Then if you remember, I told that this DNA, when you are like isolating purified, you are going to cut it with restriction endonuclease and separate it in agarose gel electrophoresis. You will cut the vector also with the same restriction endonuclease and you will go with ligase for joining it. Then if you remember, I told that whether the transformation has taken place or not, you are going to check by selectable marker. So generally the selectable marker is that antibiotic resistant gene. The example which we saw was of ampicillin. So if I transfer a vector which is containing ampicillin resistant gene, so I can just select that transformed cell by growing it in a plate which is containing ampicillin. Then I told that you can amplify the DNA, and this in vitro amplification of DNA which was there, that was done by PCR, if you remember, polymerase chain reaction. The polymerase which was used, you got it from Thermus aquaticus, right? Which was your bacteria which was thermostable. 3 steps were there: denaturation, annealing and extension. So you can make billions of copies in around 30 cycles we saw. So number of DNA goes on increasing. For that you are going to require the gene, you are going to require the primer, and you are going to require the nucleotides and the Taq polymerase. And the cycle is repeated. Denaturation, annealing, then extension. So denaturation, strands get separate, annealing, primer attaches, extension it increases the length, or you prepare that DNA and then you get multiple copies. Then we saw that I am going to produce it on a large scale, and when I am going to produce this alien product on the large scale, I am going to require a fermenter, I am going to require a vessel. And I told that if I am continuously supplying a nutrient medium and I am continuously removing the fermented broth, the cells are remaining physiologically active and the amount of the product which I am producing is large, more. I can have 2 types of reactors we saw. One was a stirred tank reactor, other was a sparge tank reactor, sparge stirs tank. So here it was stirring, it was made of a vessel which was a curved base, and there were ports you saw to check the pH, to check the what you call as temperature, to check the what you call as pressure inside, and there was foam breaker also once the foam is formed, so that can break the foam. So proper mixing was ensured. You can also have a sampling port from where you can actually take the samples and see whether everything is going good. Here in the sparger one, I told you could supply oxygen and this agitator could actually mix it and it could provide proper oxygen at a very high level. And purification of this product was called as downstream processing. Once this fermentation is done, I'll purify the product, and that was downstream processing. And then I told it goes through clinical trials, through various testing, and I told that this purification and testing of all the products, it is different. All the best for a NEET exam. Right? Thank you.